Good afternoon, everyone. Thank you so much for joining us for today's Lunch a la Carte, a monthly meeting of the CART Fund, where we get to hear from one of our previous or current researchers who have been funded with a grant from Coins for Alzheimer's Research Trust. A couple of quick announcements. If you missed this meeting live, it, along with all of our meetings, are added to our YouTube channel. So please share it with those who could not join us live. Please share it on your social media pages and encourage others to subscribe to our YouTube channel. All of the dates for future meetings between now and next spring are all on the calendar. You can find those in the DAC-DB calendar by selecting multi-district events, or you can simply find them on the CART Fund website by going to cartfund.org and then clicking on calendar. And right there, it'll give you the dates every month along with the Zoom link. But if you register in advance, just as a reminder, you will get an email notification thanking you for registering. And then you'll get another one on the morning of the event that will have the Zoom link and another reminder for you. So just a few tips there to make sure you can always find us online. Past District Governor Tammy Mosteller is coordinating a new Rotary Club that will focus on Alzheimer's research. And we are very excited about this cause-based club. If you know of folks who might be interested in learning more about that, please reach out to her and she will be happy to get in touch with you. We want to say another special thank you to everyone who has signed up so far as members of our recurring donor program, which is named the Roger Ackerman Circle. We are very grateful for donors of all sizes. Of course, major donors make a huge impact in our organization, but our everyday donors of $5 or $10 a month or more also make a huge impact for us. And we want to say a special thank you to everyone who has done that. I'm going to allow past Governor Tammy Mosteller to take over now, and she's going to introduce our speaker for the day.
Okay, Dr. Puglielli is a faculty member in the Division of Geriatrics and Gerontology in the Department of Medicine. And prior to joining the faculty at UW-Madison, he was an instructor in the Department of Neurology at Harvard Medical School and Massachusetts General Hospital. His research program focuses on molecular mechanisms of neurodevelopment and neurodegeneration. Honors bestowed upon Dr. Puglielli include being named as an outstanding scientist of the 21st Century International Biographical Center of Cambridge, UK, a National Academic Affairs Excellence in Teaching Award, which I must say, Dr. Murphy last month had that same Excellence in Teaching Award, and he was outstanding at bringing it down to a regular everyday level. So I'm excited to see how Dr. Puglielli does the same. A new Investigator Award from the UW School of Medicine and Public Health, the Bestow Award for Sustained Excellence in Basic Translational Science, and 2 Merit Awards from the Department of Veterans Affairs. He's a member of the Society for Neuroscience, American Society for Biochemistry and Molecular Biology, Federation of American Societies for Experimental Biology, the Royal Biochemical Society, the Royal Chemical Society. And Dr. Puglielli lectures in neurobiology, aging, and Alzheimer's disease courses and seminars. He is a regular permanent member of the National Institutes of Health, Cellular Mechanisms in Aging and Development Study section. So we welcome Dr. Puglielli. He's also received a CART grant in 2010. So thank you, Dr. Puglielli.
Thank you. It's a great pleasure to be here, and thank you for the introduction. I don't know where you find all that information, but I'm glad you found it. Okay, so I'll try. I try to make it as lay as possible. I don't know if I'm gonna be successful. We do hardcore biochemistry. Sometimes it's difficult to translate that. And I promise I will stay within 30-40 minutes. So there are 2 concepts that I will introduce. First is what these ATACs are. It stands for acetyltransferase. And second, what proteostasis means. So we'll start with can you see my okay. So I'll start with proteostasis. It's a fancy word which basically means protein homeostasis. And I want to remind that the cell makes proteins, and we continuously make protein. They're mainly done in the endoplasmic reticulum or in the cytosol. Some of these proteins will fold correctly and some will not. So you can imagine you're making a table or a car. Some of these cars will come the right way, some will have deficit problems. And so you need some sort of a quality control assembly line to select these proteins as you make them. Correctly folded polypeptides, those that came out the right way, they must be retained and allowed to go where they're supposed to go. So for example, if they're made in DR, in the endoplasmic reticulum, and they pass quality control, they'll go to the Golgi apparatus, and then eventually can go to the cell surface or even secreted outside. The unfolded or misfolded polypeptides, those that came out the wrong way, they obviously need to be degraded for two main reasons. One, because they came out the wrong way, so they will not be able to do what they are supposed to do. And second, because they came out the wrong way, they tend to aggregate and become toxic. And so if these proteins are made in DR, for example, they will need to go to the Golgi apparatus and to cell surface if they make it all the way. So George Palade, who received the Nobel Prize in 1974, he was the first one to recognize that most of the ribosomes in mRNA are attached to the DR membrane. Ribosome mRNAs, they make the machinery that is used to make proteins. So they attach to the DR membrane, which means that DR is the primary location to make new proteins. Some of these proteins, even though they're made on the DR surface, will be released in the cytosol and become cytosolic proteins. Some of them, however, will insert into the membrane of DR or go into the lumen of DR and become cargo proteins, so that if they survive quality control, they can be shipped out to the Golgi, complete maturation, and go where they're supposed to go. So in essence, DR, the endoplasmic reticulum, has been engineered by nature to make proteins and to make them well. And so it needs a very efficient quality control assembly line to select the proteins that came out the right way from those that came out the wrong way. And so with that in mind, between 2007 and 2010, which is right where we got the CART Fund award, we discovered the ER acetylation machinery, which is simplified in this cartoon. You have first a membrane transporter, 81, which moves acetyl-CoA from the cytosol into the lumen of the ER. This is a very important biochemical step. Acetyl-CoA is made here in the cytosol, highly charged, so it lacks water. Is unable to cross a lipid bilayer, the membrane of the DR, because obviously likes water. It's sort of mixing water and oil. And so nature invented this transporter, this channel that moves acetyl-CoA from the cytosol into the lumen of the DR. Here, it's used by two acetyltransferases, ATS1 and ATS2. We discovered this in 2010, which will move the acetyl group from acetyl-CoA donor of the acetyl group to the lysine residue, the acceptor on the protein. So this is a simple biochemical reaction that I schematically show here. You have the lysine residue. The acetyl-CoA is used by the enzymes to acetylate the amino end. So it's just a 2-carbon unit. It has very small post-translational modification that has a huge impact for life. So this free CoA is released, but it cannot accumulate in the RNA, so it goes back to the cytosol through the same door. So 81 is sort of a revolving door, using what we call an antiporter mechanism. Acetyl-CoA goes in, free CoA goes out. And so why did nature go through all these trouble and make this complex machinery? And so we made 16 mouse model. I should actually say we have 4 more in the pipeline. So we are about to hit 20 mouse models to study why we need the ER acetylation. And it boils down to this short sentence. The ER acetylation machinery regulates proteostasis within the ER and secretory pathway, regulates protein homeostasis, which is essential for life. And it does that by regulating two biological functions associated with the ER. One is engagement of the secretory pathway. The second is reticulophagy. What does engagement of the secretory pathway mean? So it turns out that we discovered that glycosylated and non-glycosylated protein can be acetylated into the lumen of the ER. The ATAses only acetylate correctly folded nascent proteins. Acetylated proteins move to the Golgi apparatus, non-acetylated do not. So what does that mean? In this cartoon, you have a correctly folded polypeptide. It interacts with the oligosaccharide transferase, becomes glycosylated, it came out the right way, it's recognized by the ATAs, it becomes acetylated. And because acetylated, it can move to the secondary pathway, can move to the Golgi and then eventually go where it's supposed to go. This misfolded polypeptide came out the wrong way. And because it came out the wrong way, it's not recognized by the ATAs, it's not acetylated, and it's stuck in the ER. So that simple 2-carbon unit, the acetyl group, is sort of a stamp of approval, quality control. This protein came out the right way. I can live with this. I'll send it where it's supposed to go so that it can do what it's supposed to do. This guy did not come the right way. I do not want it. And so I have to retain in the ER. However, these polypeptides cannot just stay forever in the ER. It needs to be disposed. This is sort of the trash. You can't just keep this trash in the kitchen all the time. At some point, once a week, you have to put it outside so that you can actually get rid of it. How do you get rid of it? And I'll go in more details later on. It turns out that if you shut down acetylation in the ER, the cell dies. It doesn't simply die. It dies by autophagy. This is from Greek, means self-digestion. The cell self-digests itself. Autophagy is a process through which the cell can digest organelles or proteins that are sick or unwanted. And so what we discover is that the acetylation of a specific protein, autophagy protein 9A, which is this guy here, regulates the disposal of toxic protein aggregates. If acetylated, this protein is blocked on both sides in the ER by chireticulin, on the cell surface, in the cytosol by HSPB1, and is unable to interact with FAM134B. Under this condition, there is no reticulophagy. If ATG9A is not acetylated, it can interact with FAM134B, and there is activation of reticulophagy. Reticulophagy means ER-specific autophagy. So basically, if I need to digest something, I'm sending that machine only in the ER. I'm not digesting everything. It's sort of having the trash, you put outside just the trash, not the stuff that is right. So it boils down to this cartoon. The ER acetylation machinery dynamically adjusts the balance between the engagement of the secondary pathway by correcting for the polypeptides and the removal of toxic protein aggregates through reticulophagy. So it's a balance. You make proteins, some come out the right way, you send where it's supposed to go, and what came out the wrong way, you remove it through this machinery. Why is all this important? And we go now from biochemistry, we go to humans. It turns out that there is a duplication of AT1 in children. Duplication means instead of having 2 copies of AT1, one from mom and one from dad, there's got an extra copy. Sometimes an extra copy, sometimes it's 2 extra copies. So there is more AT1 in these children. They develop intellectual disability and progeria. Intellectual disability is easy to understand. Progeria is another fancy word, which is Latin and Greek. It means in favor of old age. Pro in favor of, geria is geros, old age. So it's something that mimics an accelerated form of aging, but it's not the normal aging that we all go through. It's sort of a pathogenic form of aging, and I'll show some picture later on. It turns out that duplication of these enzymes, the ATAses, are also associated with the same disease. So we try to model this in mouse so that we can study them. And this is one of the first mouse that we made. You can see this is a mouse that has been engineered to overexpress AT1. It would mimic the children with direct duplication of AT1. They are born with Mendelian ratio, so everything goes well through development. But as you can see, after 1 month, they're smaller. 2 months, they look very sick and very old. We recently made mice with overexpression of only ATS1 or only ATS2. They also develop this progeria phenotype. So this is sort of an accelerated aging. It's just not normal aging. It's a sort of a disease form of aging. I don't want you to go through all these tables, but I just want to stress a few things. Lifespan, instead of living the 3-4 years on normal mice, they just live about 4 months. Lordokyphosis, cardiomegaly, osteoporosis, hair loss, defect wound repair, sarcopenia, rectal prolapse, systemic inflammation. So they all have symptoms that are characteristic of a segmental form of progeria. So this is very important for human biology and pathology. And so we focused on the 2 enzymes, ATS1 and ATS2. And you understand why we specifically focus on this enzyme later on toward the end of my talk. First, we try to obtain structural biochemical data. And we discovered that these enzymes work the same way. They use a bisubstrate mechanism, which means that at the same time, they bind to both substrate, the donor of the acetyl group, acetyl-CoA, and the protein, the acceptor. And what we discover is that very similar. The acetyl-CoA pocket is basically identical. This is the acetyl-CoA, and this is the structure of the protein. Acetyl-CoA again in the structure of ATS2. And this pocket is stabilized by these 3 amino acids. And if we mutate these amino acids, we block the ability of the protein to acetylate. So we have a target now. And if we target this pocket, we would be targeting both ATS1 and ATS2. So if you think down the line, if I want to develop compounds that inhibit these enzymes, if I target this pocket, I know that I will be blocking both ATS1 and ATS2. And this will become more relevant later on. However, there is another pocket where the protein needs to insert to be acetylated. And in ATS1 is normally under closed conformation. In ATS2 is always in open conformation, which means the protein can go in and out every time. In ATS1, this pocket needs to be open. It turns out that it's opened by self-auto-acetylation of this lysine residue. More acetyl-CoA. In the presence of acetyl-CoA, ATS1 becomes acetylated, this pocket opens up. So more acetyl-CoA, more opening. Less acetyl-CoA, less opening. Why is this important? In humans, skin fibrillas, so these are biopsy from skin, normal humans, from children up to 90-something age, the levels of AT1 go up as a function of age, not those of ATS1, ATS2. Which means as we get old, we have more AT1 on the surface of the R. We are pumping more acetyl-CoA into the lumen of the R, and we're getting activation of ATS1. And if you have hyperactivation of the R biology, this is what you get. So now this is therefore relevant, which means the ATS1 is probably my best target if I want to deal with disease associated with aging in humans. But can we really live without one of these ATAses? Can we block the ATAses? And so to answer that question, we generated mice with a genetic disruption of ATS1 or ATS2, knockout ATS1, knockout ATS2. This means that we engineer this mouse so that it doesn't have ATS1. This one was engineered not to have ATS2. The mice are normal lifespan, which means the ATAs can compensate for each other, and you can live with just one ATAse. We also discovered they have slightly different functions when it comes to reticulophagy. ATS1, which is this guy, is more relevant for reticulophagy than ATS2. So that's now fits and it becomes very important. You see that later on. We went down to the biochemistry, to mass spectrometry, and to see whether there is all complete overlap, and the overlap is just minimal. These are the we can actually track each lysine residue that has been modified through mass spectrometry. It turns out only 19 sites are shared. So the great majority of the sites are not shared. So these 2 enzymes originated probably as a single duplication event. Lower organisms have one enzyme. For example, Plasmodium falciparum has just one ATAse. And so if you shut down ATS1, Plasmodium falciparum dies. And that's the parasite that is responsible for malaria. Now, in malaria, they're trying to find these compounds to block the ATAse and kill the parasite. In mammals, we have 2. So throughout evolution, we were able to nature developed a second copy of ATAse. But they evolved to play slightly different roles. We also know that mutations that disrupt translation of either of these ATAs, ATS1 or ATS2 in humans are not associated with disease. This study was done in normal individuals between the age of 50 and 80. No disease association. They have a stop codon, which means they cannot make either ATS1 or ATS2. So there is a small group of individuals throughout the planet that live a normal life, no disease, and they only have one ATAse. We also know from the mice that knockout of one of these ATAs does not cause a significant phenotype. This means now that I can target one of these enzymes and presumably without side effect, okay, for therapeutic purposes. And this is what is summary of the last few slides that I presented. We could target ATAs to improve the ability of the ER to digest and degrade toxic protein aggregates. And before we move to translation, I have to introduce these 2 concepts, ERAD1, ERAD2. ER-associated degradation type 1, ER-associated degradation type 2. Type 1, you have the single misfolded polypeptide. It came out the wrong way. However, it did not aggregate. Because it's 1, the ER can push it through the lipid bilayer to the membrane back into the cytosol, where it's disposed of by the proteasome. But then you have polypeptides that are sticky, that tend to aggregate. And because they aggregate, they are not able to cross the lipid bilayer. So what? But they cannot accumulate there because they're toxic. So what the ER does, surrounds this complex with a membrane. So it buds off, it makes sort of a budding element, these vesicles that will bud off from the ER, become completely independent from the ER. And this is disposed through autophagy, self-digestion. This is what we're targeting, okay? And because of this essential biological role, autophagy is becoming now an active target for biomedical research. There are many diseases, and I'll talk briefly about that are characterized across lifespan, that are characterized by the accumulation of toxic protein aggregates. So if I can engineer compounds that improve our ability to digest this, to remove this trash, we can resolve many diseases, not just one. So again, many diseases across lifespan, which means in the young and the old, are caused or characterized by the aberrant accumulation of toxic protein aggregates. And one of them is obviously Alzheimer's disease. So the question now, after all these about 15 years of work, can we target the ATACs for translational purposes? The answer is yes. We spend time, and we owe this also to the CART money that we received about 10 years ago, we were able to identify compounds. We were able to gather structural information on the enzymes. So now we can study these compounds, see where they bind within the enzymes. And these are the papers where we're able to develop, show that this can actually be done. There were a couple of proof of concept that I wanted to have in my hands before moving forward. Proof of concept 1. Can I use these compounds and target the ATAs in model of hyperacetylation? This is the most relevant model, in my view. You take a mouse that has increased acetylation in DR and develops a disease. Can I block the increased acetylation in DR and rescue that disease? And you know that I do have that mouse. We generate that mouse. And the answer to that question is yes. This is the mouse that is increased acetylation in DR. Brothers or sisters, we give the compound. You can see that it's a sharp rescue of the phenotype, no disease. This is my genetic control. It tells me, yes, the compounds are actually mimicking the genetic control. This is the lifespan. The mice, this guy died a week later. All these colony of 81 overexpressing mice died fast. In 4 months, they're all dead. If I give them the compound, they have normal lifespan. So I can really prevent this phenotype. There is one problem for these mice, for this study, is that the compound 9 was given at weaning, which is the time when the mice stop using milk from the mother and start eating solid food, more or less at 3 weeks of age. But that's not realistic. The patient goes to the doctor because he's sick. So can I rescue this phenotype when he's already developing? And that is done here. We take mice, 2 months of age, no treatment, they die. I give them the treatment, they don't die. This is mouse, the wild type, completely normal. This is the AT1. You can see the difference in the phenotype. This one has increased acetylation, 2 months of age, like the brother that doesn't have increased acetylation. It looks very sick. I take this mouse, I give them the compound, 2 months later, it's completely normal. 8 months, completely normal, and it doesn't die. And this is happening both males and females. Proof of concept number 2. Can we target the ATAs in a model of ER versus cytosolic prototoxicity? I told you at the beginning that we can make proteins on the ER, but also on the cytosol. If we make them in the cytosol, they misfold in the cytosol, obviously they will aggregate in the cytosol. But if we make them in the ER and they misfold in the ER, they will aggregate in the ER. And I told you that this machine is relevant only for the ER prototoxicity, not for the cytosolic one. So we use 3 mouse models. A mouse model of cytosolic protein, Huntington model. Huntington protein is a cytosolic protein. A model of ALS, amyotrophic lateral sclerosis. SOD is a cytosolic protein. And the model of Alzheimer's disease, APP, is an ER protein. It's made in the ER. It turns out that these compounds cannot resolve the phenotype of the Huntington phenotype, of the ALS phenotype, but they do resolve the phenotype of the Alzheimer mouse. And this was published in 2016. And I'll show you some picture from that. This is a mouse with Alzheimer's disease, controlled diet. You can see the plaques, this high magnification. These plaques are very similar to what you find in humans with Alzheimer's disease post-mortem. These mice were engineered to develop Alzheimer's disease. But if I give them the compound, this is what happens. The plaques are completely gone. And whatever is left here is very small. These mice, because of this neuropathology, they die faster. So at 14 months, 50% is dead. You can see 50% at 14 months is dead. But if I give them the compounds, they live normal lifespan. So I can rescue Alzheimer's disease in the mouse model of APP. There is a third proof of concept which we're working on right now. There are genetic diseases where you have a mutation that causes the product to misfold and cause a specific disease. But then you have people that have an extra copy of the same protein, same gene, just normally folded, and they also develop the same disease. A model typical is Charcot-Marie Tooth disease. Is one of the most common, which has peripheral neuropathy. And so you have children that have a mutation that causes the protein to misfold, but also children that have an extra copy that make more of that protein correctly folded. And we're working on that. So the answer so far, can we target the ATAs for translational purposes? Is yes. The biochemistry tells us that we can target each ATAs in a selective fashion. Now we have the tools to select compounds that go after one enzyme or the other. The mouse tells that inhibition of the ATAs or genetic disruption of one ATAs is viable. They live a normal lifespan. The human data tell us that one of the ATAs seems to be enough. They also tells us that ATAS1 is probably my best candidate. So now it took us about 10 years to go from the biochemistry down to this data. Can we speed things up? And so we've been working as a pipeline to develop a possible pipeline to go identify these compounds fast, check them in silico, which fancy word to say on the computer, and select the best one so that we can go to the animal. So first, we need the pipeline to develop compounds so we can do that. We gather enough structural information that we can actually see where the compound binds. It binds in the pocket where I have acetyl-CoA, or it binds in the pocket where the protein binds. So I can block either both of them or just one of them. I can measure the affinity, and so choose what are my best compounds to target both of them or just one of them. And I can build now a table where I'm told where I can select those that seem to work for both enzymes or those that seem to work just for one enzyme, like compound 11, for example. Now I need a mouse where I can test these compounds fast and very easily. And we have this mouse that I showed you before. This is an example for compound 9 and compound 19. This mouse is 2 months old. I start the experiment, it's completed, you can see it's sick. I give the compound 9, 1 week later, it already looks better. 2 weeks later, it looks much better. 3 weeks later, 4 weeks later. I don't need to wait 16 months or 1 year or 2 years to see where the compound is working. This is a compound 19. This is another mouse, 2 months old, it looks very sick. Give the compound, 1 week later, already looks better. 2 weeks later, it looks much better. 3 weeks, 4 weeks. So in 2 weeks, I know where the compound is working or not. And so, hopefully I didn't go too fast. We now know that we discovered a machine that regulates essential functions of the endoplasmic reticulum. It regulates the ability of the ER to ship correctly folded polypeptides to the secondary pathway. And this is important for every cell, but it's particularly important for neurons, okay? Because you have to ship stuff from the cell body down to the synapse. And remember that my brain is connecting in a way to my foot. And so I need this fast communication. So I really depend on the secondary path to have control. So it's very important for neurons. The second, it allows the ER, it regulates the ability of the ER to dispose of toxic protein aggregates through this specific mechanism. I've been giving this talk, but these are the people that do the actual work. It's a long list of people. Some of them even moved to South Carolina, for those that are in South Carolina. And this is a Christmas party, the last Christmas party that we had at my house. And you can see the large group of some of my paintings that I try to do in the free time. And thank you. And I'll be answering any question.
I am a community educator for the Alzheimer's Association and a 30-year nurse. So that's why they always do I do the introductions because no one else can pronounce all these things. Just thinking. So this is the first time I've heard of the folded proteins. I've not heard this part before. We talk a lot about tau and beta amyloid proteins. Are these tau and beta amyloid proteins, are these an example of these folded proteins you're talking about?
Yes, well, so tau is a cytosolic protein. When hyperphosphorylated tends to aggregate, it forms these insoluble aggregates within the neurons. The A beta is also aggregated and aggregates both inside and outside in the amyloid plaques. So in Alzheimer's disease, you have the tau pathology and you have investigators who work on tau, and you have the amyloid pathology, and you have scientists who work on the A beta. This machinery, as I show you, it doesn't work for cytosolic proteins. So it's not going to work for tau. It's going to work for the APP, because APP has to insert into DR, and then the A beta can aggregate inside or can be aggregated outside.
Okay. So, and I don't take a lot of time, but so am I understanding correctly that when this the AT1 level gets high, that's when you're having all these advanced signs of aging. However, you can give it under if it's ER, not cytosolic, but when you give it this compound, it seems to reverse the type of aging from an elevated AT1. Is that what I'm understanding?
Correct. So I'll share again. I'll share again the screen. Can you see this?
Yes.
Okay. So this is AT1. Because I get more acetyl-CoA inside, I'm activating one of these enzymes, AT1, so I get more acetylation. The compound is going to shut down here. It's not going to shut down AT1. I can't do much with AT1. If you have an extra copy of AT1, you do have an extra copy of AT1. Unless we discover a way to do genetic therapy and eliminate that extra copy, you can't do much. But the enzyme, you have a compound, and the compound is given with a food. Is able to go to the liver. We have to measure that. It survives liver because liver has been engineered by nature to metabolize any sort of junk that we eat, almost any sort of junk. And so it survives liver. It goes to the blood, is able to go to the brain, can cross the lipid bilayer, goes in the cytosol, gets into the ER, inhibits these enzymes. Okay. That's why it took us 10 years just to find those compounds, because it's quite complex.
So the compound basically removes the improperly folded proteins.
Yes, the compound blocks these enzymes.
This is fascinating.
It blocks one of these enzymes and therefore stimulates the reticulophagy. And so now you're shifting the balance, still sort of eliminating the trash every day instead of every week. You're more efficient eliminating this trash from the cell.
It doesn't have a chance to build up.
Yep.
This is interesting. Okay.
How is the enzyme administered to the mice?
The compound, well, so we synthesize the compound, we outsource that. So chemistry. Then it comes to us. We do quality control to make sure that the compound is what it is supposed to be, the purity. Then we ship it out to a company that will make that in a diet for mice, solid diet. Then the diet comes back and is in the food. And so we give the food to the mice. It's measured so that we know exactly how much they're making. Obviously, to go that step, it seems simple now, but to go that step, we had to synthesize the compound, send it to a facility that does pre-formulation of the formulation development. Basically says the compound is stable, is not stable. It's stable in water, is stable in solid food. Can be absorbed if it's in water. Can be absorbed if it's in food. What kind of vehicle it needs. It can be absorbed. It is metabolized or not by liver. It's stable. All that stuff. And everything worked well for a couple of compounds. Some of them were very promising. but didn't manage to survive all these evaluation. And so basically means we have to modify chemically to make it good enough. And so we worked only on the compounds that were selected, were able to survive this pipeline. And then those compounds then can be administered to the mouse. Solid food, we tried to mimic as much as possible the real situation. Imagine yourself, you get you go to the doctor, get the pill, and we take the pill orally. Yeah.
This is fascinating. I think we should all congratulate you first on your patience.
Thank you.
Because this is a very intricate process you've been through, and it's fascinating that the results are fascinating. They really are.
Okay, thank you. Yeah.
I have another question. I'm going to
Yes.
piggyback on what Rod said, which I love that question, Rod. I didn't even think about how is it administered. How close are we to actually putting some of this into practice with humans?
That's why we developed the pipeline. So it took us 15 years almost to get down to 4 compounds. We started with a library of 15,000 compounds. I said we went from 15,000 compounds, we went down to 4 compounds. Of this, 2 work very well. One is so-so, one doesn't work. So from 15,000, we end up with 2 good compounds. Unfortunately, 95 to 97% of all compounds that work in the mice fail in humans because we are not mice, or we should say the mice are not humans. So the fact that the compound can be absorbed by the mouse does not mean it's going to be absorbed by humans. The fact that the liver does not metabolize this compound in mice does not mean that the human liver is not going to metabolize this compound. So far, we have worked with natural compounds, not synthetic compounds. I prefer, since I have a medical training as well, I prefer natural compounds because they come from the natural environment, okay? They're not synthetic. In theory, during evolution, we came across these compounds that tend to be less toxic than any weird compound that we can make in the test tube. That's the only reason. So we had to develop a pipeline to make more and more compounds. That's why we worked to make these pipelines that I show at the end, so that we can actually test new compounds and eventually go identify a pool of compounds that we can test in humans. There are a few tests that we can do in vitro to see what these compounds are predicted to be toxic in humans or not. And from these, we know that 2 of these compounds would be safe in humans. But again, it's a conditional. It would be safe in humans. You have to do some other tests to actually go to humans. So it's a long process.
I do have one more question. And
Yes.
if I'm taking up someone else's time, please raise your hand. What have we done to determine that this doesn't work in humans? Like, have we given this to humans?
No, there are in vitro systems, cellular systems that you can use to test whether these enzymes, these compounds would be toxic. There is one test to see whether there are 2 major problems that need to be tested. One is whether these compounds can bind to DNA, okay, and therefore can cause cancer. We can do with an in vitro test. We can see what these compounds can cause change in DNA. The answer is not. Second, many compounds, when you go to humans, they don't give problems to mice, but humans can give you heart arrest, cause ventricular fibrillation. So it's heart arrest. So it's very problematic. And to solve that, there is a test that you actually do in yeast to see whether that would inhibit the enzyme that is responsible for that in the yeast. We do that too. And they both survived. So there are these in vitro tests that you can do preliminary to predict. That's why I say predict whether these are going to be toxic or not. And then obviously there is an entire pipeline to bring compounds from the mouse to humans that needs to be followed.
Are there research, or does your lab do research on trying to develop a compound for the cytosolic, like that doesn't typically respond to this? Are they also studying that cytosolic for like the?
No, we are not
How-
studying that.
Okay.
We're not doing that, but there are other labs that are working on that. We're interested in ER proteostasis, not in cytosolic proteostasis.
Okay, I was just curious if we had something. Did I understand you to say also that Huntington's and did I hear ALS were also two diseases that also causes increased AT1?
No, these are two diseases where you have increases aggregation of proteins in the cytosol.
Okay, okay,
For us, there were controls.
okay, gotcha.
So the question is, are these compounds specific enough? Okay, because there is acetylation in the nucleus, there is acetylation in the cytosol, and there is acetylation in the ER. We developed these compounds for the ER. Are they truly selective? Okay, so that's sort of something that you need to know. You have to see where the compound is going to give you side effects. So in our case, there was a control.
Okay, gotcha.
It's a proof of concept.
Gotcha.
It is truly fascinating, and we do appreciate your time and your commitment to this project and to your research and to the betterment of trying to find a prevention, treatment, or cure for this disease that we are all so equally passionate about. So we appreciate your participation in the CART Fund and your continued support as well.
Thank you.
We thank everyone for your participation today and hope that you will share this video with friends and coworkers and fellow Rotarians after we get it posted to our YouTube channel, and that you'll invite them to join us again next month as well.